Okay, so the next experiment, experiment 12, is the endospore stain. And the purpose is to just go over the differential stain of endospore stain. Um, so that's the next in the line of differential stains after gram stain. So it differentiates endospore formed cells from non endospore formed cells. It's not forming, like, it's not the ability to form it, because if you have a bacteria that has the ability but hasn't formed it, it's not going to stain the endospore because there is no endospore to stain. So it differentiates cells that already form the endospore from cells that have not formed the endospore. Um, and endospores form in genetically capable gram-positive rods when nutrients are depleted, and they're said to be in what's called a cryptobiotic state, in that there's no metabolism occurring, so they're not breaking down anything or building anything. They're very just null. Um, and the sporulation process, like how the spores formed, is on page 53. Um, and it takes you step by step on how the endospore forms. So I'm not going to go into that too much. Um, yeah, I'm not going to go into that too much at all, actually. So what we did in okay, well, the purpose of the endospore is to protect the bacteria from anything that happens while it can't grow and germinate. So obviously, endospores are very, very tough, from what you've learned in class already, and uh, they just kind of go into this endospore state, and then once the nutrients and the environment becomes good, they'll recognize it, and then they'll sporulate again back into a vegetative state. Um, and so this is important for this is important. I remember from I think my TA said that botulism was an endospore forming thing from and if you've ever worked in a restaurant, you'll know that like botulism is like a very important thing that they consider. Um, they always refer to like the hazardous time of like sitting, and that's when of like food sitting, and that's when botulism can like um, grow from its endospore and become like bad, um, and get you pretty sick. But yeah, and so that's the important. So it's a very dangerous thing, kind of, and autoclaves are kind of designed to beat endospore forming cells. So that's why they have such high pressure and high heat, is to make sure it destroys all cells, including ones that even form endospores. So but what we did in the experiment was we took we took a endospore forming cells. We took B megatherium and Bacillus megatherium and Bacillus sphaericus, and we did an aseptic technique on it, and we put that on a slide, and then we watered it because it was from a solid medium. And then what we did was we cut out like those paper towel squares, and then we flooded it with malachite green, and that's the one where you had like a can boiling, and it had like the net over it, and you put it on there, and you let it sit over the boiling water for like 6 minutes, and then we washed it, and then we counterstained it with safranin. And I should have gotten a picture of it. Let's go. Um, let's see. So you get pictures like this, in that the endospore, it's green, and then Non-endospore stuff is the safranin colored of like that pinkish red, and so that's the whole purpose of the endospore stain. This is kind of my weakest lab in explanation because I didn't really learn the endospore stain stuff very well when I took the class. I mean, I learned it well enough to understand it, but it's been a while. Um, it is important to note that this Bacillus megaterium is central forming endospores, and this Bacillus sphaericus is terminal forming endospores. Um, but other than that, I'm not ask the doctors if you know if you're confused about this after you read it in your lab manual. Um, but I'm not going to go over too much about how the endospores form just because that's going to take way too long. Um, so read your lab manual and then ask someone if you're still confused after that.